to Hubert Institute. I'm Masoumeh Ali Mohammadi, and what I want to do in this short video is an overview of the endomembrane system. As you know, there are different types of organelles within eukaryotic cells, and some of these organelles are part of the endomembrane system. This is an eukaryotic cell, and the endomembrane system is a group of membranes and organelles which work together in order to modify, package, and transport proteins and also lipids. So what membranes and organelles I'm talking about? First of all, the cell membrane itself. So the endomembrane system includes the cell membrane, nuclear envelope, and the endoplasmic reticulum, or ER for short, and Golgi apparatus, and also lysosomes, which are originated from the Golgi. All of these membranes and the membranes around these three organelles, are phospholipid bilayers. So if I were to zoom in right over here, it really is a bilayer of phospholipids. So it would look like this. You have your hydrophilic heads pointing outwards and your hydrophobic tails pointing inwards. And it keeps going. So you have a layer of two, or you have a bilayer, and I should say a bilayer of phospholipids. That's going to be true for the cellular membrane, that's going to be true for the outer membrane of nuclear membrane, and also for other cellular membranes. As I said before, these components are functionally related and work together. So in the following, I want to talk a little bit about the endoplasmic reticulum and the Golgi apparatus and their roles in maturation and transportation of some kinds of proteins, including membrane proteins and also the secretory ones. The endoplasmic reticulum is a network of interconnected membranes which are forming tubes or flattened sacs, as you see here. So the ER really has two regions, the rough and the smooth. This depends on ribosomes, which are protein synthesizing factories of the cell. This is a free ribosome right over here, and this is an attached one which is attached to the outer surface of the ER. These attached ribosomes giving a rough appearance in electron microscopy. So this region where you have many attached ribosomes right over here and looks like flattened sacs is the rough ER, or ER for short. And then there is another region without ribosomes, which is more tubular, called a smooth endoplasmic reticulum, or SER. So let's compare these two types of ER in this chart. The first difference is based on the presence or absence of ribosome. RER has it and SER doesn't have it. And the other is about their structure. SER is more tubular than RER. And what about their functions? RER is the site for receiving proteins, while the SER is the site for synthesis of lipids and estrogen. And as you see, both structures are involved in post-translational modifications in which proteins can be chemically modified in order to get mature or get ready for its destination. Let's go to previous slide and see the Golgi apparatus or Golgi body. It has two components. One is flattened membranous sacs, which are called cisternae, right over here. As you see, they are placed one on top of the another. And the other is the small membranous vesicles. The cisternae of the Golgi apparatus have three distinct regions: the cis region nearest to the nucleus, and the trans region closest to the plasma membrane, and the medial region lies in between. And we have a lumen here. Lumen is the interior space of the Golgi and also the ER. These three parts contain different enzymes and perform different functions. The Golgi has several roles. It can modify proteins like adding carbohydrate groups, and it also concentrates, packages, and stores protein before they are sent to their cellular or extracellular destinations. At the end, just as a review, let's see how these organelles of endomembrane system work together in order to transport a secretory protein to its destination, which is outside of the cell. The rough ER is a key for starting this process because the attached ribosomes are actively involved in protein synthesis and translate mRNA directly into the lumen of the RER. These newly synthesized proteins right over here undergo several changes, several chemical changes, kind of post-translational modifications. And then these mature proteins are safely transported into the cis region of the Golgi apparatus by some sort of vesicles. The Golgi may further modify them, adding some addressing tags indicate where eventually they should go, and finally the fully matured proteins, which are enclosed in vesicles bud off from the trans membrane of Golgi apparatus and are transported to their final destination, the extracellular environment in this case. That was an overview on the endomembrane system. Thank you for watching and bye for now.